Reviewed your knowledge of genetics, and you understand that the difference in between the nucleotide sequence within a gene and how is that connected to the amino acids sequence within the protein that the gene coded for, then we can go forward and learn how these sequences can be very useful in comparing different organisms and how are they related to each other in our pursuit of phylogeny and cladograms and evolutionary trees and so on. So we already know, while reading Making of the Fittest, that some genes are very highly conservative. They're so important to the survival of the organism that has that gene that they are not, if they are mutated, whenever a mutation occurs, most of the time is detrimental and the organism doesn't survive. So how is that affecting the gene within evolutionary time? Is that gene preserved and we call it immortal, or is that gene fossilizing and we call it a fossil gene? So go back to that concept and compare and contrast immortal genes versus fossil genes. So again, the question here, these very highly conserved pieces of DNA or particular genes, are they immortal or not? So when we compare that, we can tell the difference in how are these organisms related to each other. And we'll find out that many organisms have a very similar sequence of nucleotides in between genes on very different groups. So that explains a little bit why our genome can be so similar, more than 20% or maybe even 28% similar to rice. Okay? So in general, if you look at the sequence of amino acids in a protein of close relatives, you will see that it's very similar. And the longer they have differentiated, the longer the time they have differentiated from each other, then the more different mutations have accumulated. So let's take a look how this looks like. So we have a group of 20 organisms here. All the honey creepers have been lumped in one, so it's just one picture of it, and then you see two more other birds, and a mouse, and a bear, and a fish, and a plant, and a louse, and yeast. So, and we're looking at a piece of cytochrome B, and that is a mitochondrial cytochrome B. Let's see, you can see this. So, cytochrome B. Okay, so this is the protein. So this is a sequence of this particular protein, mitochondrial cytochrome B. And the question to you, is this a very highly conserved gene or not? And why? So if you chose one versus the other, can you support your hypothesis? So if we look at this comparison of the amino acid sequence in between these 20 organisms, we'll notice that all the honey creepers have exactly the same sequence. There is no red in this whole line, and the red stands for mutations. If we compare the honey creepers with the other two birds, right, the song sparrow and the finch, or in particular this Gull Island finch, you will notice that there is one mutation and then maybe two mutations or three on the other one. And then we move to a mammal, a mouse, and there are more mutations, and we can see that the mouse is more similar to a bear than to the fish, and so on. And the farther we go, so in between the honey creepers and then this particular here on the bottom to this baker's yeast, or Saccharomyces cerevisiae, which happens to be the same with brewer's yeast. But this particular unicellular eukaryotes, so yeasts are eukaryotes. It's very different. It's a fungus, so it's in the kingdom of fungi. So it's very different to all the animals and the plant that are listed here. And you can see the difference by accounting for how many mutations can happen. Okay. So, but how do evolutionary biologists, and in general biologists, understand that list of just letters, right? So you have all these letters here. What are these letters standing for? I'm telling you here that it's comparing amino acid sequence in this particular mitochondrial cytochrome B protein. But what are these letters standing for? So we know that there are about 20 amino acids, and hopefully you remember the names of these amino acids from studying genetics, and in particular when you talked about translation from genetic sequence of nucleotides in a gene to the sequence of the amino acids in a protein. So you looked at which sequence of, which codons, which three, the sequence of each three combinations of the 4 nucleotides in DNA and actually in RNA from the messenger RNA on the genetic code, which 3 of these nucleotides account for alanine or arginine or asparagine or glutamine or all the way to valine and so on. So originally, to shorten the names from the whole long name of each of these amino acids, a group of 3 letters were used because it's easier to remember that ALA stands for alanine, and ARG stands for arginine, and VAL stands for valine. But then who wants to write this with 3 letters for every amino acid? Even now we are doing it computerized, but there are more chances for mistakes. So eventually these 3 letters code has adapted to be a 1-letter code. So now it's a little bit more difficult to remember that R starts for arginine and W starts for tryptophan. There's no W in tryptophan. But if you use this often enough because of what you're doing, then you will remember that D stands for aspartic acid and so on. So that's how that list was made. Now, if we look at comparing sequences of nucleotides, we're looking at comparing DNA or pieces of DNA or pieces of genes, not necessarily a whole gene, sorry, the other direction. So we know that within a cell, if it's an animal cell, we'll have DNA both in the nucleus and the mitochondria. If it's a plant cell, it will have DNA in the nucleus, in the mitochondria, and the chloroplasts. And those pieces of DNA will be very different in between the nucleus versus mitochondria versus chloroplasts. So this is another good opportunity for you to take a moment, turn the video off, and try to recall what do you remember from previous biology classes in between those 3 organelles. Nucleus is considered an organelle, versus mitochondria and chloroplasts. How did they evolve? All organisms have mitochondria, or what organisms don't have mitochondria, and what organisms have or don't have chloroplasts, and so on. And what is their size comparative to each other, right? It turns out that nuclei are the largest, followed by the chloroplasts, and then by the mitochondria, and these are the 3 largest organelles found in a cell. And these are the 3 organelles that carry DNA and genetic information. But there are very big differences in between the nucleus eukaryotic DNA versus mitochondrial and chloroplast DNA. So again, I want you to review that, and please ask me questions if you have. But we already know that most of the time mitochondria, and it turns out that also chloroplast DNA, it's inherited through the maternal side. And it makes sense because the eggs are much larger versus sperm. So sperm has mitochondria, but only in its neck, because it needs the energy to outcompete all the other sperm cells to reach the egg first. But only the head that contains the nucleus enters the egg. The rest of it is left out. So very small chances, occasionally it happens, but most of the time, all the mitochondrial DNA that you have in all your cells is on your maternal side. So very interesting phylogeny studies can be done by following mitochondrial DNA. Another reason why mitochondrial DNA is more beneficial sometimes to phylogeny and phylogenetic studies, it's also because it's haploid, and there is no recombination. There could be mutations, which we're interested in, because remember molecular clocks. If there is an accumulation of neutral mutations in the mitochondrial DNA, we can determine, so according to how many mutations, how far away those two sister groups have differentiated. The more neutral mutations have accumulated, in this case in mitochondrial DNA, the longer the time that those two groups have diverged, so differentiated. So what do we have here? First of all, is it comparing DNA sequence or protein sequence? And you can only see ACGT, which are the 4 nucleotides found in DNA in all life on Earth. So it's comparing 10 types, the 10 types or the 10 species of Hawaiian honeycreepers. And whenever there is a red letter, you can see that there is a mutation. So since they are the same group, they are related. We noticed that when we looked at the previous comparison of amino acid sequence in the mitochondrial cytochrome B protein, there were no differences in that particular gene, so in that particular protein, but in this case, we're looking at the DNA and not necessarily of the same gene that controls that particular, protein in cytochrome B. And we can see that there are some differences between them. And we definitely know that there are differences between them. They were allocated different names and they are in different species. And they, if you look at the morphology comparisons, you can tell that they have adapted to different modes of feeding, right? So this particular one has a very hard beak for probably cracking seeds. And this one here has a much thinner beak for collecting nectar in this particular bottle brush, and so on. So those are comparisons that lead to cladograms. So this particular cladogram here, or evolutionary tree, phylogenetic tree, was made on the basis of the DNA sequence that we just seen. And I want you to take a moment so you can actually see the names of these Hawaiian honey creepers, and I added the pictures on the side according to their order, right? So these 5 and these 5. And I don't know if you noticed, but some of the Hawaiian names are as difficult, in my opinion, to the Latin names or scientific names. The good news is you don't need to remember either of them. But I do want you to take a couple of minutes and take a look at this cladogram and compare the two sister groups, right? So you have two sister groups here. So there's the one on the bottom, the 5 on the bottom, and then the 5 on the top. And then it's rooted to this more farther away group also of Hawaiian honey creepers. So take a couple of minutes and let me know what you find interesting. And the one thing that I noticed, and of course there are lots, the more you look at them, the more you notice, is the fact that when they were assigned names of genera in this case, according to the morphology and anatomy, so the comparisons between morphology and anatomy of their cranial features and characteristics, these two, so Hemignathus virens versus Hemignathus monroe, were assigned the same genus, which means that at the time they were considered to be very similar. And now we see that this particular one is rooting the two sister groups that are here. So they have evolved farther away. So DNA sequence or protein sequence that is based on the DNA sequence. So new tools of comparing biochemistry allowed evolutionary biologists to revisit the cladograms and evolutionary trees that were done just through morphological data. And in this case, it's very important to determine which groups of these Hawaiian honeycreepers store the highest genetic biodiversity, which probably would be the most important to save. As you know from reading your textbook, all of those, but one particular, the species on the bottom right here, that it's not immediately endangered of extinction. All the others are endangered because of habitat destruction and invasive species that humans have brought in to the Hawaiian Islands in the last 1,000 years already since was discovered. They were discovered from other continents.